Hello, so good afternoon, everyone, and good morning to Paola, actually, and to many others that they are on the other side of the ocean. So today is really my great pleasure to introduce Paola Arlotta to our seminar series. She's without any doubt one of my science heroes, but not only, she's also a great colleague and a great friend. So a little bit of history about her career. So Paola is Italian, and she studied biochemistry in Trieste, in Italy. And then she got her PhD in molecular biology in UK, where she focused on studying high mobility group proteins. And then she made a big move. So she went to Boston at Harvard Medical School in the lab of Jeff Maclis, where I think she got really inspired about her future work as well. And there already she made a big contribution in understanding how the brain develops. And this was, and it is, the core of her present research. So then since 2007, Paola is a professor of stem cells and regenerative biology at Harvard. And recently, I think a couple of years ago, she also became chair of the Department of Stem Cells and Regenerative Biology. She's not only principal faculty member at Harvard, but she's also a member at the Broad Institute. And of course, she has collected a lot of prizes and awards, but what I would like to emphasize is that she's investing also a lot of energy in supporting, encouraging, and promoting the career of young scientists, and especially young female scientists. Paola's work Paola's lab works on a variety of subjects, but fundamentally she's focusing on mechanisms underlying brain development in the cerebral cortex, but also at the specifically of how different subtype of neurons comes together and integrate into functional circuits. And their work span from cell fate to reprogramming to pure brain development, but also to understand how this is integration in the circuits in the end. And to achieve that, she's using a couple of model systems, so the mouse developing brain, but also since a couple of years, she's very much well known for studying human cells by using brain organoids. So I think with that, I will let Paola talk about her work. Today, she will present her work on, and the title is Understanding Brain Development and Disease from Embryos to Organoids. So Paola, thank you very much for being here today. And yes, so please share your screen.
Thank you.
We are very looking forward to your talk.
Thank you so much, Silvia, for the kind introduction and for the invitation. And so nice to see many of you. Thank you. I'm going to go ahead and share. Let's see. On your screen.
I see it.
And then see my slides. All right. Can you see my slides?
I see it perfectly. So
could you see the pointer moving a little bit?
Yes, I see the arrows. The only thing that I see is you are screening your entire screen. Maybe if you can put it down, but otherwise in the middle of the slides.
Which one? This one?
This one, exactly. If you could close this small icon, exactly, so that it's not in the middle. Perfect.
Something like that. Okay, That sounds great. All right, thanks so much again, everyone, for having me here today. As Silvia alluded, my lab has been interested for a while in understanding sort of the molecular rules, the molecular principles that during development control the generation of the great diversity of cell types that are present within the mammalian central nervous system, and how these cells then come together to build higher order structure and connectivity as seen in the brain. And a lot of our work uses the mouse as the mouse embryo as the model system. But recently, we have really wondered how much of this beautiful process of brain development that the embryo executes so perfectly could actually occur outside of the context of the embryo in stem cell-derived 3D human brain organoids. And I find this exciting and inspiring because it offers a window of experimental opportunity into the human brain, into a process of human brain development that we have never had an opportunity to study. So I'll share with you some of that work today as well. Okay, so the part of the brain we're interested in is the cerebral cortex. As all of you know, the cerebral cortex contains a very large diversity of cell types. They come in many different flavors, and most of the cells are really produced through an extended process of development that occurs in utero, followed by many years for human beings of experience-dependent maturation of the cells, the neurons, the circuit that are generated in the embryo. And it is this very complex and prolonged process that really allows the cerebral cortex to build the circuits and networks that underlie very complex type of behaviors. Now, if we think about the cell types that are present within the cerebral cortex, as I mentioned, they come in many different flavors. And if we concentrate on the neurons for a moment, although one could classify them in two large buckets here, the excitatory long-distance projection neurons and the inhibitory locally connected GABAergic interneurons. So each one of these categories contains many, many different classes. The projection neurons are the excitatory neuron, just to set the stage, that connect the cerebral cortex to all of its long-distance intracortical and subcortical targets, while the interneurons connect locally within the cerebral cortex with themselves and the excitatory neuron to together form the cortical microcircuit, which is, of course, extremely important for cortical function. Now, this diversity of neurons, excuse me, also interacts with a great diversity of glia. Again, they come in three major buckets at least, but each bucket contains many types and many states, as we're understanding now from a variety of single-cell RNA sequencing effort. And what my lab has been sort of interested in is really how does this diversity of cells come about? How do the cells interact with each other to build the cerebral cortex? So what I wanted to do today is to start by showing you some data about a general approach that we're taking to understand how cell diversity in cerebral cortex is produced in vivo using the mouse embryo, as I mentioned, as the model system. And then in the second part of my talk, we go to this question, how much of this highly orchestrated process of cellular diversification that one sees in the embryo can actually occur without the rest of the embryo. So outside of the embryo in stem cell-derived brain organoids. Just as a sort of to set the stage for the topic, this is a topic that we have been interested in for a very long time. Understanding how all of these different classes of neurons, for example, shown here in the many different colors, how do they develop? How do we go from progenitors that have, in theory, the potential to make many different cell types to generate this diversity of cells? And in the past, anybody who wanted to ask this type of question was faced with the difficulty of actually studying individual classes of cells from a tissue that contain so many different types. And so we had to figure out ways to sort the cells from the tissue, isolate individual classes of neurons, for example, and then use the molecular profiling of the moment to really understand what were the transcriptional states of the cells. And this kind of cumbersome type of work extended through development allowed us to a certain extent to get at what the codes of transcription factors, or some, I would say, of the transcription factors that label individual classes of neurons, and in some cases to identify what some selected transcription factor really do to the development of this cellular diversity. But I think that because it's fair to say that the field advanced relatively modestly with this kind of approach, largely because it's very, very difficult to purify these cells, to do them in large numbers, to really study them using this type of technology. In addition, it's very clear that no single neuron types, for example, of the cortex develops in the cortex in isolation. Its development is always together with all the other cell types. And so development is really a process of orchestrated differentiation of many different cell types that influence each other while this process is occurring. And so despite the fact that, I'm going to move this someplace else, maybe here. So despite the fact that a lot of progress has been made, we still don't, we have some fundamental questions that have not been answered. how is the cellular diversity of all the cell types of the cortex controlled and governed? How are the switches in among lineages really occurring, and how are they regulated? And then more importantly, perhaps, how do these cells interact with each other to build the higher order structure of the cortex, the layers, the circuit, the connectivity, and so on and so forth? And I'll mostly concentrate on these two first questions today, but if you're interested in assembly and how pyramidal neuron diversity, for example, affects the development and function of oligodendrocytes and myelination, we had a recent paper that is now out on this specific topic. I won't go into the science today. So these are the questions that are still there, I feel, today, and they're mostly still there because the technology we were using was not the right type of technology to take a systems biology approach and look at every cell at all times during development. This has been now. Okay, so we have recently addressed this problem directly, and this is the work done by Daniela Di Bella in my lab in collaboration with Hasan Habibi, and he's a postdoc in Aviv Regev's lab. So all this work was done in collaboration with Aviv's group, and they decided to really collect using technology that now is not only available but commonly used of single-cell RNA-seq and single-nuke ATAC-seq to collect very well-defined regions of developing somatosensory cortex at every single stage of development from E10.5, shown here, every day, all the way to P4, with a little bit of space between P1 and P4 here. And then use single-cell RNA sequencing or single-cell ATAC-seq and in situ transcriptomics to really profile molecularly this cellular complexity. And when you do something like this, you finally get sort of the map of all the cells at saturation. All you need is to sequence enough cells to make sure that even cells that are presented at very low percentages within the tissue could be sampled and statistically analyzed. And this is what we always wanted. We wanted sort of a dynamic map from the beginning to when the majority of cell types are made. All of the cells are present within the cerebral cortex, and you can see how they diversify and increase in types as development progresses as expected. It's relatively easy to classify these cells from their molecular profile in the different classes that we know quite well. And so with this data set in mind, let me give you in a few slides some example for what you can do with this kind of data. One thing that one can do, which I find very exciting, is to organize the cells, given that they are collected at multiple developmental time points, into a pseudo-time lineage tree. And so this is a tree where at the tip of the tree, you'll find the differentiated cell types. Here I'm indicating some of them. And then the start of the tree is a single node that represents the most undifferentiated progenitor cells that we were collecting. And so if you walk through this tree through time, then you also walk through lineage bifurcation decisions that progressively lead to specific cell types. And what this really means, given that this tree is constituted by single cells, so every little dot here, it's hard to see because there are many of them, constitute one cell and its molecular profile, which means that if you now start from here, from the progenitor, and you walk through single cells all the way, for example, to, let's say, a callosal projection neuron of layer 2-3, then you're reading the molecular recipe that controls the formation of that cell. All of the genes that at a point or another in the life of the cells were expressed as this specific neuron type was made. And you can repeat that for every single differentiated tip in your tree, and you're going to have that full recipe. I'm excited about that because so far we only had sort of snapshot in time of when of specific cell types. Now you can have it for every single one of them. Perhaps more important to understand the principles of cellular diversification is that this kind of data and this kind of tree also allows you to identify the switch codes. These would be cluster of genes, probably mostly transcription factor and DNA binding proteins that at a specific point of bifurcation within the tree, let's say here, switch number 2, separate the right branch from the left branch from the mother branch. The idea would be what changes really in gene expression soon before this lineage bifurcation occurs, and can we then take those genes and begin to understand whether they control indeed the switch. And so if you do this across multiple of the switches as identified in the tree, here they are shown, then you can identify transcription factors. For example, concentrate now here again on that switch number 2. You see transcription factor that distinguish number 2 from number 1, so from the mother branch, and also the left branch from the right branch. So the branch that would lead to the deep layer neuron versus the branch that would lead to the upper layer neurons. These switches are very interesting because now, although they are not the totality, of course, of the programs that define a cell type's control its development, they are among the most likely genes for controlling that specific point in time. And so I'm excited to see whether we could use them to understand how lineage bifurcation decisions across all of these different cell types are controlled. And this becomes much richer and much more about mechanisms rather than simply expression of genes if you build this atlas not only in single-cell RNA-seq space, but also in single nucleotide-seq space, where one can actually look at DNA accessibility regions and begin to correlate gene expression, transcription factor binding in those regions with regulation, basically, of gene expression. And so we have built, in addition to a single cell RNA-seq tree, also a single nuc attack-seq with less ages than the single cell RNA-seq, and found that indeed the two correlate quite well in terms of regulation. And finally, in a tissue like the cerebral cortex, where in addition to having a great diversity of cell types and a temporal order by which the cells are built, there is also a lot of migration of the cells to progressively expand outward the tissue. And so we figured that we would have an opportunity not only to identify cell types and gene expression profiles, but also to relate those to specific positions within the cortex. And so what we did here was to collect the same embryos, the same pieces of cortex, and then use a slide-seq, which is an in situ transcriptomic methods, to identify cells and gene expression profiles physically within the tissue. And then use a new algorithm that was recently developed by Aviv Regev's lab to relate our single cell atlas that I just described to you with the position of the cells by slide-seq inside the tissue. And what you can do with that is sort of shown here. For every single cell, you can locate them in space and also understand in that specific position at that specific time, cell X expresses a certain set of genes. What this kind of profile gives you, for example, are these transitory states, these transitory molecular states that migrating cells go through as they position themselves from the germinal zone up to the cortical plate, for example. These were pretty elusive states to identify in the past. So we're excited to now have them. And this perhaps is shown here, where if I look at all of the cells, this is an E15.5 slide-seq, one can actually follow in gene expression space and in the tissue the transition from intermediate progenitor cells to neurons as they migrate to the cortical plate. So sort of in general, and there is much more data here, but let me give you sort of the take-home messages. We built the single cell transcriptional and chromatin accessibility atlas of the developing cortex, and we feel that that has given us now quite a bit of molecular information to begin to understand how globally this process of cellular diversification may occur in cerebral cortex. From this kind of data, you can extract a lot of information about principles. For example, and it's just an example, one finds that diversification of neurons occurs mostly postmitotically and not at a level of the progenitors. There is a lot of information that one gains about the progenitors and their fate, and so on and so forth. but also it's possible to associate gene expression with position and get those elusive transition states that cells acquire as they migrate. Most importantly, I think, we get the molecular recipes and the codes of switch for all of the cell types, terminal differentiated cell types that one can identify within the tissue. And this is quite important because this is the bridge to understanding what these genes do and therefore how these different classes of cells are made. Now, this is all nice and good. However, in order to fulfill that sort of prediction or that will, one has to also figure out how to turn a massive amount of molecular information into biological understanding, basically, of the process that we're trying to understand. And this is why, as we were building this resource, Ashwin Shetty in the lab, a very talented postdoc in my lab, and then Xinjin in the lab as well, in collaboration with Feng Zhang and Aviv Regev, decided to set up a high throughput functional screen in utero to be able to screen for potential function of very large number of genes in a small number of experiments, again, taking advantage of available technology. And so what they do here is to generate very large guide RNA libraries and inject them in utero in a Cas9 animal, and then let the embryo develop, so they deliver them to the progenitors, and then use single cell sequencing to begin to understand what each one of these perturbed genes does to specific cell types. An initial part of this work is published, so I'll go really quickly and in a schematic manner here, but just to give you a sense of what kind of pipeline one could implement in this space. So this is work led by Xin, who was interested actually in genes that are associated with disease of the cerebral cortex, and in particular ASD. And what she did, she selected a relatively small number of genes, 35 of them, made a guide RNA library that had 2 guide RNA per gene and a fluorescent protein plus a barcode that would uniquely identify the perturbation, and injected those as a library through viruses in Cas9 embryo A12.5, so at the beginning of corticogenesis. She then let the cortex develop and collect tissue at P7, followed by single cell transcriptomic and analysis of the data. And what you do in contexts like this is that you get a lot of single cells out. Here is an example of the cells being simply bucketed in very large buckets, astrocytes, microglia, astroglia, and so on and so forth. And now you can select specific clusters of cells, let's say oligodendrocytes, and then ask what genes, not just single genes, but groups of genes, so cluster of genes, modules of co-regulated genes, change and in what cell types. And these modules of genes could reflect all sorts of biological processes, from cell states, cell types, developmental events. And then you can combine this information and say, okay, in a cell where a certain modules of genes change, what kind of perturbation did that cell receive? And if you combine that, you get this type of heat maps where you have here all of the perturbed genes. Each column is a perturbed genes, and then each row is a gene in the module that has changed. And you can see, for example, the 2 perturbations, CHD8 and GATA-D2B, produce a downregulation of a module associated with OPC differentiation in this specific case. So I hope I gave you a sense for how you can start for a massive amount of molecular information, collect it through development, and then filter it through high throughput functional screen to get at perhaps a beginning of an understanding. So just in summary from this part of my talk, corticogenesis is complex. We've always known that the cortex form as an orchestrated development of many, many different cell types, which are marked by thousands of genes and that proceed in ways that we don't fully understand. Scalable technologies, I think, has changed the landscape for how we can address problems as complex as understanding how diversity of cells emerges in cerebral cortex. And these become, I think, very powerful if you combine them with scale functional phenotyping to screen through a lot of genes for a first pass. Of course, you still then have to go in and really study these genes deeply, but at least the codes could be perhaps identified. And I think this changes the approach. Maybe it would allow us to take a systems biology approach to understanding how cortex is made. So this work is very rewarding because it happens in a system, the embryo in utero, that very reproducibly makes the brain, and it's an experiment, a very good experimental system. However, there is a limit to what we could understand about development of our own cortex, of other cortices, by studying the mouse. And we've always been fascinated with understanding better how the human cerebral cortex develops, except we have never had a way to really study it, because the process of human corticogenesis occurs in utero, and we will never have access to that kind of process. So then the question emerges, how much of that beautiful process of development that we see in utero in vivo can be reproduced without embryo, perhaps from stem cells in human brain organoids. And I think this is exciting because, for a variety of reasons, if we could have a good experimental model of some aspects of development of the cerebral cortex, and a lot of things could really happen. The model is needed, as I mentioned, because there are fundamental differences between species, and while the mouse is absolutely fundamentally important and we're learning a lot from it, there are also aspects of development of the human brain that cannot be studied in a mouse or in any other organism. We will never have access to proper human development because it occurs in utero. And most importantly, for those of us who are interested in neurodevelopmental disease or in neuropsychiatric diseases, we cannot ignore the fact that human genetic studies of the last perhaps 5, 6, 7 years have given us a pretty complex genetic pictures of these diseases. And the picture says that these are polygenic illnesses whereby a large number of variants scattered across the genome would produce a certain likelihood of development of the disease. And so for diseases like these, where the entirety almost of the human genome is needed to understand what genetic does to the brain, then this cannot be modeled in a mouse. And so we need human experimental models of the brain. And this was our entering into the field of human brain organoids at this point a few years ago with the work of Giorgia Quadrato, very, very strong postdoc in the lab. She now has her own lab at USC. And Giorgia began by addressing what I think it's a very important question that relates to the speed of development of the human brain. If we want to study cellular diversification, for example, within the human cerebral cortex, then we need to accept that this tissue will develop really, really slowly, and it will do so in vivo, as we know, but it does so in vitro as well, and we can discuss that. And so she built on pioneering work by Madeleine Lancaster and Jorgen Oblich, Yoshiki Sasai, showing that one could start from pluripotent stem cell and generate brain organoids. At that time, when the first studies were published, these organoids could only be cultured for a relatively short amount of time. And this is when Giorgia came in and asked whether perhaps this culture could be extended, this development could be extended substantially in the future. And long story short, she succeeded at that. And in her paper showed that she could grow organoids in bioreactors for about 9 months without major problems of cell death in the center of the organoids. And it is her organoids that were still growing now in the lab to really understand what the boundaries of this process of corticogenesis outside of the embryo really are. So this was important for us because not only they survive for 9 months, they continue to add new cell types and develop. So with this kind of milestone in place, then she began to ask what is really inside a human brain organoid. And this is a brain organoid grown for 4 months, stained for some markers for cortical progenitors, as well as markers for neurons of the cortical plate. And as developmental neurobiologists, one thing you can see immediately that it doesn't look at all like an endogenous human developing brain. But if you look a little bit more closely, you can see that there are areas of the organoids that resemble some features that you also see in vivo. For example, look here and in many other places where you have this ventricle cortical-like structure, above which a layer of early progenitors is set. These are the apical radial glia. Above them are the intermediate progenitor cells. And then the outer layer is the cortical plate-like structure where multiple type of neurons are located. We've done a lot of single cell sequencing to define actually the cellular composition of these layers. And so this was exciting to us because it was the first picture at that time that told us that there is enough cellular complexity in here perhaps to really get to the bottom of how they develop and what they do. However, we very soon hit a wall, and the wall, or the big elephant in the room, was that very differently from the embryo, these organoids were quite unreproducible, meaning that every time an embryo makes the brain, it always makes the same brain, and that's why it's the perfect system. But every time we were making an organoid, it would be a little bit different from the next organoid, and so very hard to use them as experimental systems. And this is when Silvia Velasco in the lab decided to take this problem on and set up in the lab many different protocols for organoids, and ultimately settled on this protocol shown in picture here, described here, which was a modification of an original protocol developed by Yoshiki Sasai, and asked a bunch of questions. Could this protocol be giving organoids that can grow for long term so that generational cell diversity can occur? Can it be established reproducibly from multiple stem cell lines? And most importantly, can we have every organoid undergoing the same process of development? And I will not, this is published, so I will not go too much into this, but allow me to show you at least one slide of this work, because this was the point for us where we felt that indeed we could begin to use these organoids to answer some biological questions. What you see here at the top are pictures of organoids grown over time to 3 months or 6 months. And what you see below it are 2 experiments, examples of many more experiments. You can see here about 500,000 single cells. So this data, it's actually supported by a lot of experiments across 70 single organoid, multiple batches, many different stem cell lines. So 2 separate experiments where we took 3 single organoids and sequenced them separately with single cell RNA-seq. The first thing that you see is that in both of these experiments, where I merged together the cells from all 3 organoids, they both form the same diversity of cell types. And some of the cells are listed here, which are appropriate cells for the cerebral cortex. So this was great because it told us that the same cell type can be made in 2 separate experiments. And in fact, these were even 2 different stem cell lines. But what was really important is that when we looked at the individual organoids, each one of these organoids produced the same diversity of cell type. So that this was not just a mix of the 3, is that each one of them would produce the same cell types, with a level of reproducibility actually that when it was calculated compared to the endogenous tissue was very similar to the endogenous tissue. This was really rewarding because, again, elevated this model to an experimental model that could then be used. And I'll share with you some unpublished now data showing that not only by the time these organoids reach 6 months or 3 months, they have reproducible cellular composition, so they're similar to each other within a batch and across batches, but also that the whole process of development of these organoids is reproducible. And so this is work done by Amanda and Anna. Amanda is a bioinformatician in the lab, shared with Aviv Regev's and Joshua Levin's lab, and Anna is a postdoc in my lab. And what they did was to build a single-cell RNA-seq atlas of development of the organoids, sampled at multiple times of development that we chose based on what we knew about the cell types that were made at specific time point. And they did this extensively through, again, many single organoids, multiple time points, many batches, and a lot of single cells. And this really allows us to say that not only late in development, but also earlier, if you take individual organoids and you look at the cell types that they make, they're very reproducible in the cells they have and the timing also of when they make the cells. And this extends throughout the entire process of formation of all of the cells that we can make within the organoids. Not only the cells are made reproducibly, but the proportions of cells within each single organoid, every column here is an organoid, and the colors are different cell types. I'm just showing you some here, was actually quite reproducible. And in fact, you would have a very similar picture if you did the same thing with single individual endogenous cortices from a mouse or from a fetal human cortex. This made us dig a little bit deeper into molecular programs. So the genes and programs and modules that would control or label specific classes of cells through development across different experiments. And so I'll show you here an example for two major classes of neurons of cortex. These are corticofugal projection neurons, and these are the callosal projection neuron, again, produced within organoids. And what you see here are heat maps where at the bottom you see many different types of stem cells that we started with. So each column is a differentiation from a different stem cell line. And we're looking just, let's say, the corticofugal. And so you can see that the signatures that define every row, is a gene. The signature that define these neurons are very consistent throughout experiment and across the cell lines. And the same thing here for callosal neurons. Not only that, when we compared corticofugal to callosal, we saw that they had very complementary type of signatures that were labeled by genes that we know are specific markers for corticofugal versus callosal neurons, which again gives us a sense that we can study not only the cells appear and they're made, but they appear and they're made through a consistent developmental process that relays on similar set of genes, which then can be studied. And this is the first time that we're watching human cellular diversification really occurring. Now, how real is this? I know that there have been a lot of claim about how similar the cells that are made in organoids are to the endogenous cells made in the embryo. And we need to continue to remember that these are reductionist in vitro systems, that's for sure. But they are pretty good, at least in this specific protocol with the differentiation that we have set up. And so what this slide shows is a work that we have done in which we have trained a classifier. So it's an unbiased classification of the cells. So we have trained a classifier on a data set of human fetal cortical cells produced by Dan Geschwin's lab, and then use the classifier to ask the classifier to classify the cells of the organoid so that to understand how they would relate to the endogenous one. So here are the endogenous cells with the names that the authors gave to the cells. And here are the cell types that we gave them. And we found that there was really a very high correlation between, assigned by the classifier of the organoid cells to the correct type of fetal cells. We also did the opposite, where we trained the classifier on the organoid and went to the fetal and got similar results. In our hands, we don't see, and we dug a little bit deep into this, major metabolic changes, with the exception of two cell types among 14 different cell types that we studied. And so the classifier and the matching of the cell was not affected in this specific culture conditions by the metabolic state of the cells. So these, in my opinion, tell us that it's time to use the organoids to perhaps explore biology that we have always wanted to explore and never could, keeping in mind that they're still experimental system in vitro, that's for sure, but they're the only system that allow us to study human genetics and human biology. And I'll end with literally 3 or 4 slides of a recent study. Is in bioRxiv, so you can see the totality of it, where we began to ask whether these human brain organoids could be used not only to understand normal development, but also to get some insights of human neurodevelopmental disease. And to do so, we decided to start with ASD and ASD risk genes, and in particular, single genes that, if mutated, are associated with a risk for autism. And ask what these genes then would do to the neurobiology of the human brain analyzed within organoids. ultimately to get a question like what cell types are affected, what developmental trajectories, if any, are affected, what happens to the circuits that are produced in this genetic context. And this is a massive amount of work that took really the work of 4 different postdocs in the lab. Amanda, again, as a bioinformatician, and then Silvia, Bruna, and Martina doing the more biological organoid work. And they started with 3 genes, which are some of the highest hits in autism risk genes. KMT5B, which is also known as SUV420H1, P10 and CHD8. They took IPS lines so in this case, we modified the lines to carry a heterozygote mutation in these genes. And this was a specific choice because we wanted to understand this independently for a moment from the genetic background, and then grew these organoids, either isogenic controls or those that would carry the mutation, and then use a variety of single cell and bulk approaches to understand whether phenotypes were present. The first thing that the organoids allowed us to do is to show that all of these 3 genes, CHD8, P10, and SUV, were indeed expressed within the organoid, so likely their function could be studied here. And as expected, they were expressed in a variety of different cells. So they're expressed in many different classes of cells, and I can tell you they're also expressed across many different stages of development of the organoid. Other genes that came out of the genetic studies were also expressed in the organoids, and some of them with very much cell type specificity. So the first thing that was obvious as we were growing these organoids is that in all 3 cases, a high percent of the organoids grown showed larger size. So this is an organoid carrying a mutation in SUV420. This is a control, and the mutants were always larger than the control across the 3 mutations with different degrees of phenotype. And this was exciting because many of the patients that carry this mutation also display macrocephaly within a larger segment, in particular of the cerebral cortex. And so it was a way for us to see that we were perhaps able to model even something a little bit more complex and detailed than this. And indeed we could. Lots of work summarized it just in a few slides. Let me give you the gist of it. We grew organoids again, either carrying a heterozygote mutation in each one of these genes or their isogenic control. And what you're looking at here is an example of one experiment where we grew this organoid for a month in bioreactors as well as later ages, but the phenotype occurs at this stage. On top row is 3 individual organoids from the isogenic control, and at the bottom the mutant. First thing that you see is that the organoids are very reproducible to each other. And this is the only reason why it's possible to read a phenotype, because if there was a lot of variation in terms of the cell types made and number of the cells, that would be very difficult. And what we consistently saw for this gene is that one population of cells, despite the fact that the gene is expressed in many different cell types, one population of cells, the deep layer projection neurons, were always present in larger numbers in the mutant compared to the control. And this was repeated in separate experiments where, again, here, this is another experiment where you see that this specific population of cells is expanding while all other cell types remain unaffected. This was interesting, but it became interesting when we began to look at the other mutants. This P10. P10, of course, is a gene that does a lot of different things. But one thing that we didn't know it did, at least in human organoids, is that by 3 months in bioreactor, in these mutants as well, we saw an increase in one specific population of cells. In this case, we're still neurons, but these were the callosal projection neurons, while other cell types remained unaffected. And finally, when we look at CHD8, now the phenotype became very, very obvious. And here are 3 organoids control, and here are 3 mutants. I should say it took us a long time to generate this data because we really wanted these organoids to be reproducible enough, basically, to be compared to to each other. But what you see here is that in the mutant, you have a much higher numbers of both progenitors and neurons, now in a different class. These are the inhibitory cortical interneurons. So here you have 3 genes that appeared from the genetics of autism as being high-risk genes that we couldn't quite put together in any specific obvious way in terms of convergence of phenotype, that were expressed in a variety of different cell types and a variety of different time points, and yet demonstrated a phenotype in very specific classes of cells. And the phenotype is similar. There is an increased number of neurons of a class of another, neurons that ultimately are supposed to come together within the cortical microcircuit. And this is just a repeat of the CHD8 experiment, where again, here's the control. There is a massive expansion of interneurons within the mutant. And I should say that there is beautiful work out in bioRxiv from our colleagues Gaia Novarino and Giuseppe Testa, also identifying this interneuron phenotype in CHD8. So it's very exciting that organoids are beginning to show some of the biology associated with these mutations potentially in the human brain. This is another way to look at this data, where instead of asking are there more or are there less, we're watching development of these organoids and try to understand whether there is an acceleration of neurogenesis in this context. And indeed, this is very a somewhat complicated slide, but perhaps you can concentrate here, where you see a pseudo time of development of the cells. And if you look at the very end, so among the more mature cell types, you see that in the SUV, instead of compared to the control, there are many more cells, and these are the deep layer neuron one more time. And if you look at CHD8 and consider the interneurons, by the end of this pseudo time, there are many more interneurons in the CHD8 than there are in the control. So this is exciting for many reasons, and we have done a lot of molecular work as well as proteomic work to try to understand this deeply. But fundamentally, this phenotype speaks to a level of convergence for these 3 genes that molecularly were very hard to somewhat converge. They converge not at the level of a common molecular pathway or mechanism, but at the level of a developmental process, a process of neurogenesis, which gets accelerated in all 3 cases, affecting, though, 3 different classes of neurons. What is interesting is that these are the classes of neurons, as I mentioned, that will come together in the cortical microcircuit, suggesting that possibly a higher level of convergence happens at the level of perhaps misfunction of that circuit due to these early developmental abnormalities. And with that, I got to the end of my talk. Sorry for going a little over. I'd like just to state a few things. Organoids are reductionist stem cell models. They are grown in vitro and they have all of the defects of anything that is grown in vitro, but they also have a lot of potential. And I hope with the data that I showed you, that they are able to reproduce some aspects of development that can be effectively studied in the context of normal development or disease. I show you data from 3 ASD risk genes that are converging over a common defect of early neurogenesis and possibly defects in the assembly of the cortical microcircuit, which could be really interesting from a therapeutic entry point. I'll end with some sort of more general thoughts. I think that we now have an opportunity with all this new technology to take a slightly different approach as a field to understand brain development, looking at all cells, all times, all modalities, and figuring out computational pipelines to really integrate all this information in a single picture of what this process really is. Thanks to the same technology, never like now, we have had access perhaps to the human brain, both in terms of being able to actually profile and obtain static pictures of the endogenous brain, but also through these human brain organoid models, which for as reductionist as they are still the one experimental model of the human developing brain. And I can see that the field is expanding exponentially, and I hope that in future years we'll be able to use them for more and more complex type of studies. And with that, I'd like to thank you for your attention. I think I mentioned the names of the people in my lab who did the work that I presented today. Many more work on understanding endogenous normal development in the it's the work, I think, that roots the work in organoids of understanding how our own brain develops, perhaps, or some aspects of it, or how it is affected by disease. And with that, thank you so much again for the opportunity, and I'll be delighted to take questions.
Thank you very much, Paola. I will be your audience now and just clap for you. So fantastic talk. I have many questions, but I think we will start with a question from the people there. So exactly, so wonderful presentation. So this is from Pablo M. Wonderful presentation. I was wondering if it is possible to combine brain organoids with another type of organoids that allow vascularization or mechanical constraints by skull formation to try to stimulate natural environment.
Yes. And this is something that many people are doing, I feel, in the field right now. We are very interested in mechanical forces, so we're not doing much work with vascularization, but just to answer your question, it is possible to create vascular beds outside of the organoid that then, when put in contact with the organoid, could invade the organoid and create a scaffold. And it's also possible to flow in this vascular bed things. I don't know if it is literally blood, but a variety of different things. And so it could also be a delivery method for delivering a variety of factors to the organoids, and also to improve survival in the center of the organoid. After the organoids reach a maximum size, you begin to see some cell death in the center. In terms of mechanical forces, I find that very, very interesting, fascinating. We are working in that space of really learning from the embryo how really the embryo uses these mechanical forces, both to generate multiple brain regions, but also to generate specific points of convergence of tissue where the mechanical forces can really affect even cell fate. And there are a lot of things that are, of course, missing in the embryo, in the organoid that the embryo has, that we're trying to engineer. I think that those kind of forces must be engineered. They don't self-emerge, and that's where perhaps the rest of the embryo really matters.
Thank you. So next question. Thank you. This is from Frontiera, from actually our Max Planck. Thank you for sharing your science with us. I'm wondering if you have ever tested the reproducibility of your protocol with organoids produced from stem cell line reprogrammed either from fibroblasts or from PBMCs, so from different somatic cells.
From different somatic cells. So we have not done a systematic study of that. We have compared ES cells with iPS cells, but we haven't systematically, although we have, I'm trying to remember now of the iPS, I think they all came from fibroblasts. So I guess the short answer is that we have not done that. If your iPS line is a good line that doesn't tend to spontaneously differentiate and passes quality control for being a good iPS line, chances are that you're going to get an organoid with this protocol. It's not as finicky. There are some lines that are particularly bad, as we all know. You're always going to have them for any protocol. We had a much harder time with other protocols than with this one to have lines to work. And that's all I can say. But occasionally you're going to get a line that, so unless you really love that line, you really, really need it, my suggestion would be to exclude.
Okay, so the next one is from Karan Aouya. So thanks a lot for sharing the wonderful presentation. I'm just wondering if the cells in brain organoids are mature enough to use this model for studying neurodegeneration in elderly population. Degeneration and what? Neurodegeneration in older people.
Yes. So although in theory one could imagine that So these are very young kind of tissue, right? So these it's hard to give an organoid an exact age compared to the endogenous fetal tissue, but by looking at what cells you have and the maturation state from a molecular viewpoint and also electrophysiological properties, they're probably early, late fetal, first few years of life. And so this is not the window of time when those degenerative diseases would appear. And so there was a question, there is a question still in the field of how much of that you can model. And so two things have emerged in the field. The first is that perhaps even diseases that manifest later in life in human beings may start actually quite earlier. And so even in an organoid that is young, you may catch these initial abnormalities that may then later result in neurodegeneration. The second is that there is a lot of work to these systems, especially in diseases like ALS, and to a certain extent Alzheimer's, so it's more controversial there. But for ALS, where even if the tissue is relatively young, some hallmark of the disease can appear. And this could be related to the fact that you are in vitro without the compensation that you would have in vivo. And it's possible also to sort of integrate additional insults to the culture to foster the manifestation of the disease way earlier than it normally would. So I wouldn't exclude the use of organoids for neurodegenerative disease, but I can see that perhaps the first applications would be for diseases that have a bona fide origin in development.
So another question from Mikhailo, sorry, the question disappeared while I was trying to read it, so maybe we'll come back. Maybe I accidentally deleted, so please put it back if that was the case. So one from Nicoletta Casares, fantastic talk, thank you. Cell death play a large part in shaping the maturing cortex. Have you looked at the cell death in the organoids?
Yeah, hi Nicoletta, thanks for the question. We have started looking at that. So there is certainly cell death, and there is cell death that could be classified as developmental cell death rather than simply unhealthy tissue, because it happens at stages of organoid development where you don't have much suffering of the organoid because the size is still small, because the media can penetrate, no problem. So we haven't really studied this deeply, but it, to a certain extent, is there, and so it might be possible to study. Just we've not done it.
So next question is from Elisa Moreno. Thank you, great presentation. You have shown that the proportion of neuronal subtypes is preserved across organoids, batches, and time. What about non-neuronal cells like macro or microglia?
Yeah. The cells that emerge in these organoids, because of the patterning of the progenitors, you mostly have cells derived from the dorsal telencephalon, and so you'll have all of the neurons of the cortex. There is a source of interneurons also from these progenitors, and then we can discuss that, and astroglia. And so the astroglia proportions are also maintained organoid to organoid. Microglia does not develop from these progenitors, and so a way to get microglia inside organoids is to transplant it, meaning you need to make it on the side because it just derives from another set of progenitors that you don't have here, and then coat the organoid with microglia, and they'll invade it. So we have not done that experiment, and so we'll be interested to see whether they survive differentially, but you need to add it. It doesn't co-develop with the other cells.
So the next question is, again, the one that I was starting to read before. I think someone voted and then went down in the ranking. A technical question regarding organoids and mutation linked to ASD. Proportional increase of projecting neurons could be either increase in amount of these neurons or decrease in amount of other neuron type. Did you try compositional analysis to tackle this problem? For example, such tool as C-code.
Yeah. Yes. So we have done all of their work, and really this goes to Amanda and Aviv and Joshua. We have done a lot of bioinformatic work on this. So we do know that it's an increase in neurons rather than a change in the relative proportion.
Okay, so next question. There is no name, but thank you for a great presentation. I was wondering if it is possible to model imprinting disorder using brain organoids.
Not my field. So I'm thinking, but it's something that I discussed at some point also with Catherine Dulac and others. So my answer is going to be so superficial. I almost don't want to give it. I think it might be possible, but don't take me to the bank with this, because it's really not my field. Sorry.
So we go for the next question. That is from Mateja Rybicka Tezulov. Probably not the right pronunciation. Anyway, thank you for this beautiful talk. I was wondering whether you can detect non-coding regulatory RNAs in organoids, single-cell RNA-seq data to a level that they could be used as a model for these RNAs. And if so, are these also reproducible?
Yes, we have not looked at those. So I really don't have much to say about them. I think that we can detect them, but we haven't gone deep into their analysis. We've done work with these RNAs in the embryo in the past in collaboration with John Rind, but we haven't done it in organoids. I wouldn't say, why not? You should be able to study. You can look at this model as an experimental model with the sort of the reproducibility organoid to organoid as a tissue, as an endogenous tissue. So for this process, for the process of cellular diversification.
So the next question from Zoltan Molnar, inspiring talk, Paola. In the brain, the neurogenesis is separated in space and time, and there are multiple streams of tangentially migrating glutamatergic and GABAergic neurons from areas where you have differential combinatorial transcriptional control.
Yes.
Are these cells equally represented in organoids, Hall-Retzia cells, subplate?
Yes, hi Zoltan. Very, very good question. They are there at the beginning of development, and actually for the subplate, it's harder to distinguish always the subplate, although you are the expert on this, from the early corticothalamic cells. What we don't have here is the development of areas, although you could identify some early sort of gene expression signatures of the areas that will be. So short answer to your question, yes, they are there and they are in the right proportions and reproducible among organoids, but we and others as well, just because the organoids are so young, have not really looked at those complex dynamics, many of which will happen sort of late as well. The other thing I wanted to add is that while in vivo you have this perfect also topographical organization of the cells, you mentioned the migratory streams and so on and so forth, this is not necessarily the case in organoids. They don't build complex structure. And so, for example, eventually the layers are not built. And so really identify migratory streams and in a very well-defined way, as you can very easily do in vivo, that's harder here. I think that will come with the development of better organoid models that will reflect structure and anatomy in addition to cell types.
Another question that was partially already answered. So great talk from Francis Zele. Great talk. Since the organoids were so similar to endogenous cells, but the foramen do not have microglia, does this show microglia are dispensable for cortical neural development? And have you added microglia to the organoids?
Yeah. So answer is no, 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 no. Not for a moment, I think that the microglia don't matter. I think they do matter. I think every sort of cell-cell interactions, both ways, really does matter. And so, so this goes to what are you really comparing? So what we do with the classifier, it's really try to understand how similar the fetal cells are to the human one. There might be some differences that are also due to the fact that one grows in vitro, the other grows in vivo, that don't preclude the matching, right, from the classifier. So I think you'll have to do a study that is much deeper than that if you wanted to really understand what microglia does to the neurons and vice versa. And that would require, you are correct, the transplantation of the microglia within the organoids. They don't emerge, yeah.
So I think with that, you answered the next question that was, have you tried or have you considered to xenotransplant your organoids into the mouse to provide an in vivo environment to have microglia, right?
Yeah, that we're doing. And really sort of like others in the field, get to these low level of chimerisms that would allow you to study certain aspects of the biology of these human cells that really cannot be studied in vitro. And for example, responses to sensory stimulation and things like that, that can be done in sort of in a rudimentary sort of way in the organoid in vitro, but that should really be studied in vivo. And so there is, again, depending on the question that you have, that's an option as well. The cells can engraft, as you can imagine.
So from Pier Paolo, beautiful presentation. Did you ever evaluate the temporal maturation of apical radioglia?
Did you what, sorry?
Did you evaluate the temporal maturation of apical radioglia?
Yes, and we have a project, it's still ongoing, run by Anna Osquiano, really looking at these lineages, apical radioglia to its progeny, outer radioglia to its progeny, over time, to understand how they are related lineage-wise, as well as what are the mechanisms that allow this transition to occur, how are the cells made. Very much ongoing, so I don't have much data on that.
So Louis is asking, thank you for that nice talk. I'm wondering how close this protocol can reproduce the gyrification features of human fetal brain, and whether you can see the effect in cell diversity, extracellular metrics, leading to this regulation of that feature in the P10 organoids.
Yeah, so we have done some proteomics, and we will continue to do proteomics to also understand a little bit better the protein composition of the extracellular matrix. For now, we have, of course, information about what extracellular matrix proteins these cells that we have may be secreting, but it requires, sort of additional work there. And in terms of gyrification, there are no gyri here. This is there are no gyri for a variety of reasons. One is the age of the tissue. Two is the fact that at the structural level and anatomical level, we're not quite there. It's going to be the next generation of organoids that perhaps will build structure, but I wouldn't expect gyri at this stage, too early.
So we still two questions. From Roscana, have you had the opportunity in your studies so far for pharmacological manipulation in disease models?
No, we haven't done that, but this is where we would love our work to then sort of leave the lab and enter a biotech space to begin to use them to screen. We are not a chemical screen lab, but I think that the models now with this kind of reproducibility can be a good cellular system for testing this kind of screens. You need to have a defined phenotype that you're screening for if you want that.
So one more question from Nicoletta Quesares, actually was one of my questions as well. How does you observe increase in cortical interneurons, the CHD8 mutant organoid fit with a human CHD8 mutations? What is known about inhibition in human carrying CHD8 mutations?
So very little is known in human about what exactly this mutation really does. So we know that about 75% of the patients have this macrocephalic phenotype. Okay, fine. We also know that a very high percent of patients have epilepsy. And so this is where this idea that you have an abnormal, either way, early neurogenic event that affects only one broad population of neurons, but no other cell types, that could lead to a desynchronized process of development which eventually could affect the circuit. Now, how that translates in what the patients, shows, that it's going to be hard. It's hard to tell at the moment. For now, I think we have an opportunity to really figure out molecularly and developmentally what may be going on that we didn't know before. But I think that this will fuel into studies looking at the activity of the brain in these patients in the future. Very little is known about inhibition there.
What I also found very interesting is the fact that you have in certain cases an increase in excitatory and some inhibitory, and this is really something that is fascinating. So it's just imbalance, right?
It's an imbalance. Thank you for saying it. It's an imbalance and an asynchrony at some point in development, which later on will result in probably in miswiring or in abnormal functional properties of the circuitry, whether you come in with an abnormal pyramidal neuron or you come in with an abnormal set of interneurons, you may converge in that kind of abnormalities later on.
You may. Yeah.
And now also something that can happen very early in development can actually, even if probably will get compensated. So I believe that it will get compensated through development, but still you have had that asynchrony early on, and you may see an effect later on in the circuit. It's all to be seen.
You produce more interneurons, and then the in vivo, the excitatory neuron compensate, and then the interneuron somehow, then maybe later on are the excitatory neurons that,
Correct. This is exactly the kind of then, compensation that the brain would do to compensate for that, which may lead to a circuit that doesn't work quite properly. So it's exciting to think, very speculative at the moment.
We still have questions. Thank you again. I had another question. This is no name. I had another question. How well do the organoids represent epigenetic changes during development, especially at the chromatin level?
Yeah. To answer that question, what we really need is an atlas of the fetal cerebral cortex with single cell resolution in chromatin space. So I have, we have this information for the organoids, we have this information for the mouse, but not for the fetal human cortex. So it's hard to tell how close they are to endogenous development in terms of epigenetic state. And a lot of people are working on generating this data set, which I think are absolutely needed to answer your question and many more of how similar are they.
So we have one last question. From Sara Bandiera. Hi, thank you for the talk, very exciting. It's very promising to see the good correlation you see between organoids and fetal cells. I was wondering if you check also about their functional properties. For example, do corticothalamic projecting neurons actually project towards a correct target if you co-culture them with the appropriate target organoids like thalamic organoids?
Yes. So these would require the thalamic organoid, which we may agree or disagree whether that exists. Many people, but it will exist. Inside the organoid that I showed you, there is no much, like in most of the organoid models, there is no much structure. And so you can't really understand whether they connect properly to their target. The target is not there. I would predict that if you had an organoid, a thalamic organoid, that kind of connectivity would probably happen from all that we know of how things happen in vivo. If you give them enough, if the specificity of identity is correct, and if the cells are, put together, match properly. But nobody has done it, and I think a limitation here is having good quality organoids of other brain regions as well. But the field is clearly expanding in the direction. You probably have seen beautiful work by Sergio Pasca, others assembling different brain regions that could serve as an example.
So thank you very much, Paola. I think we have answered all the questions. I would like to thank you again for fantastic, brilliant talk as always, but also the people for asking a lot of questions and generating a very nice, a lively discussion. So good evening to everyone. Thanks again, and see you next week.
Yeah, thanks everyone. Thank you, Silvia.
Ciao.
Ciao.